Humans depend for their sustenance on agriculture. Most of the energy for humans comes from plants. This facility, where you see plants going around on a conveyor belt, almost like a manufacturing plant which you see for car manufacturing, is a special place where non-destructive testing of plants is done. Scientists at the Indian Agricultural Research Institute in New Delhi are using this facility to make plants which are climate change resilient. The whole body scanning of plants using sensors, both inside the greenhouse and in the field using drones. Plants can't speak, but scientists can still understand their language. Keeping an eagle eye on plants is part of this massive facility. The facility you see behind me is the Nanaji Deshmukh Phenomens Center at the Indian Agricultural Research Institute in New Delhi. And I have with me Dr. Vishwanathan Chenuswamy. Namaskar, sir.
Namaskar.
Part of Dr. Vishwanathan's job is to make India's agriculture climate resilient. Dr. Vishwanathan, what is this facility we see behind us? Can you tell us a little bit about it?
So this centers characterize the plant health with response to various input that we give to the plant and with response to environmental factor without destroying the plant. That is, non-invasively it characterize the plant as we characterize ourselves by using MRI or CT scan. In that way it characterized. This facility cost about 45 crores. This facility was inaugurated in October 2017 by Honorable Prime Minister of India.
And why does one study phenomics?
So, what we see as a phenotype, just myself and you look different. This is because of our genetic makeup and our interaction with the environment. Similarly, these plants also have the genetic makeup and they interact with the environment every growth stages of life. So, we need to understand how this interaction leads to the plant characters, how it produces yield, how it responds to various input. For that, we need to characterize the plant during the different growth stages in different environment.
And is this important for what is happening thanks to climate change and global warming?
Yes, this is very important in reference to climate change, because climate change reduces the crop yield and reduces the crop quality. Fortunately, we have wealth of germplasm reserve which can be used to identify the genotype that can tolerate less water availability or high temperature and produce same grain yield and grain quality. So, for that this facility is very important to identify those germplasm lines which can yield better with limited input and with the climate change conditions.
Scientists like you and the institute where we are standing, premier institute where green revolution was pioneered, can you make climate resilient plants, climate ready plants for India?
Yes, we are confident that we can make. Already our institute has developed plants that can use less water to yield to the similar level to the conventional varieties that yield. We are developing genotype that tolerate to high temperature. So, I am sure that our institute will develop and agriculture scientists will deliver the plant that are resilient to climate.
Let's go and try and understand using mustard and wheat plants what plant phenomics is all about. Let's go. Dr. Vishwanathan, what do we see here?
So here we are seeing how the same wheat genotype respond to two different level of nitrogen, and here we are seeing two different mustard genotype, how they respond to same amount of water applied. So these differences in the phenotypes are characterized inside the plant phenomics center.
So essentially what you're doing is what used to take years, you're trying to do in weeks and days inside the facility?
Yes, because these studies are very difficult to conduct in the field, because when we grow large amount of genotype, the input we cannot apply specifically to each plant. Here we are quantifying the input that is applied to each plant, and then we are measuring the phenotype. So it is not necessary to repeat at different years, so we can characterize in 1 year about the plant phenotype.
Dr. Vishwanathan, let's go and take a look at the other facilities. We are inside the greenhouse where conditions of what the earth could be in a few decades are being simulated. Higher levels of carbon dioxide to increase and show global warming, and also increased humidity. And Indian scientists are conducting experiments on that. Dr. Vishwanathan, how do you simulate conditions under global warming?
So in the global warming, we expect carbon dioxide to be higher and the temperature to be higher, and there will be change in the relative humidity, but depending upon the temperature condition. So here we have facilities to increase the carbon dioxide concentration, and we have these air conditioners. They can produce heat or cool depending upon the temperature we set in.
So this is what? Carbon
This
dioxide?
is carbon dioxide cylinder.
Okay.
And then these are the humidifier. You can see humidity is coming. So depending upon the crop condition, certain condition, the pest incident will be more if we simulate high humidity. So those conditions can be studied here. And all the plants, they are placed on the conveyor system, and each car is RFID tagged. So the data of plant health is recorded in the same RFID for different climatic conditions.
Now, how much carbon dioxide can you increase here with this cylinder? Till what level can you go?
The carbon dioxide normally we keep up to about 650 to 700 ppm. That is what expected in the future climate change scenario. Otherwise, the carbon dioxide is about 410 ppm in the normal condition.
Now, these are wheat plants. What happens to wheat when the temperature increases?
So, in the wheat plant, when the temperature increases, their duration decreases. That is, it comes to the flowering quickly and it produces less biomass. And then when the heat occurs at the flowering stage, sterility occurs, so less grains per plant is produced, and the grains produced are in smaller in size, the quality is less.
And you're studying also what is happening in mustard, and what are you studying in mustard?
So in the mustard, already mostly it is grown under the rain-fed condition. So as the global warming changes occur, we expect that more drought condition, more high temperature stress condition. That is what we are examining, how the different mustard genotype respond to increase in water deficit and increase in temperature.
Dr. Vishwanathan, this was about carbon dioxide. Let's go and take a closer look at how you increase humidity. India is a monsoon country, and humidity is particularly important for India's agriculture. Let's go. What you see is increase of humidity being done in this greenhouse. Dr. Vishwanathan, one sees so much and hears about irrigation on plants and agriculture. How does humidity play a role in plant growth?
So the humidity in the air is the one that decides how much water is lost from the plant to the atmosphere. So what we call it as vapor pressure deficit in the atmosphere. depends upon the humidity. If more humidity is there, the vapor pressure deficit is less, so the plant will transpire or use less water. If humidity is less, then the deficit is more, the plant will use more water. So that's why if you alter the humidity, we can alter the rate at which the plant can use the water.
And you can look at the best genotype and the best phenotype response in this setup, correct?
Yeah, we can look at the best response, how the plant uses water, and you can see in the pots, you can see that white color beads.
Yes.
That are put to prevent the direct evaporation of water from the pot. So that is a kind of mulch. So now we measure exactly how much is the water lost only through the plant. So that is what we call as transpiration. So it is useful to quantify exactly how much amount of water each plant uses. So it is the best method. There is no other means to quantify this in the field condition.
These plants are on a conveyor belt very similar to what you see in a car manufacturing plant. These plants from here where conditions of global warming and a changing climate are simulated are taken inside for a whole body scan, almost similar to a MRI or a CAT scan for humans. A non-invasive way of trying to understand which is the best phenotype among all these genotypes. Let's go and take a look at that. We saw the greenhouse where conditions of global warming were being simulated. Dr. Vishwanathan, we can see some plants coming on this conveyor belt. What does this do and why are these plants coming here?
So these plants, as you told, they have been given the specific environmental condition where we have given treatment to this plant. Now we are going to characterize this plant, what impact that climate has made on this plant. So these plants are RFID tagged. So these cars moves on the conveyor here, and it go through this conveyor belt. It goes to the different imaging chambers. So these imaging chambers, they have specific cameras. Each chamber has different camera. So let us go and see how these plants are being imaged and the data is recorded.
What happens here inside this?
So the plants one by one, they come in this car and they enter one by one into the imaging chamber. So this each imaging unit, you see here 8 different units.
8 units are
8 units are there. In each unit, different kind of sensors are there. For example, in this unit, we have one IR camera on the top.
Which is infrared.
Which is infrared camera on the top. One infrared camera on the side. So once the plant goes inside, the image is taken from the top as well as from the side camera. And these parts also can be rotated in different angle to take different images.
And how many automated cameras like this and roughly what do they do?
So in this we have 8 different units, 8 sensors. The first sensor, it takes the thermal image. That is, it measures the temperature of the tissue. So it is very important for metabolism, and it also tells about at what rate the water is lost from the plant, that is transpiration. So this is affected by different climatic condition as well as the input condition. So we can quantify for when we reduce the water, which plant can maintain its water status and it maintain its temperature, those plants are good. So that can be done by this system. And if you see a large gap, about 15 pot, that is the dark adaptation tunnel. So in this tunnel, the pots wait there for about 15 minutes to completely make the light, whatever the ATP produced, completely it is used under that time. Then after that, it moves to the chlorophyll fluorescence system. So the chlorophyll fluorescence camera measures the photosynthetic efficiency. So inside that camera, it takes the image and it says whether the photosystems are good or any damage is there. If we move to the next imaging chamber, we have a empty chamber wherein we can put any sensors. So currently we use in that FTIR and spectroradiometer. So we collect data on the water content of the plant, and high molecular weight components that are present in the plant can be characterized in that unit. Then it goes to the visual imaging unit, where it is color imaging, just like our normal visual camera.
Like the camera which we are
shooting now. So that measures the plant growth. So it can say what is the weight of the plant. We need not cut and take the weight. It can say what is the weight. So for this we standardize once. Each crop we have standardized, we have taken the image, we measured how much the pixel area is there, that we have correlated with the actual weight of the plant. Based on this we have developed model. With this model we can predict what is the weight of the plant, and also we can see at what rate the plant leaves dry or it becomes senesced because of various treatment conditions. So that can be done by the visual imaging chamber. Then we have a NIR shoot imaging chamber, where it measures the reflectance from the leaf surface
which
in
is
the
near
infrared range. So in the near infrared range, it measures mainly the water content. So we will know, without affecting the plant, we will know what is the water content in this leaf. So this is mainly affected by high temperature or drought condition. If drought is there, the water will be less. So certain plants, they have ability to take up more water or retain more water in the tissue. So that can be measured by the NIR chamber, near infrared chamber. Then we have a sensor unit which is called as NIR route, that is near infrared route, wherein we have both visual camera and near infrared camera. So for that we use a different kind of pot, where we use acrylic pot, and in that we can take an image that tells about the root growth root system.
You can't see the root here.
Yeah, in this we cannot see. For that we use an acrylic pot which is transparent. So where we can see how much depth the root goes, and also what is the amount of water that root extract from the soil. So it measures both water extraction as well as the root growth pattern it measures. Then it goes to the next imaging unit. The last two units are hyperspectral units. So in that the imaging cameras, two cameras are there. One camera measures the reflectance from the plant from the 400 nanometer to 1000 nanometer. So the other camera measures from 1000 nanometer to 2500 nanometer. So in these two cameras, speciality is that it gives reflectance from the plant at each nanometer wavelength. So any light, when it is falls on the plant, and it is some light is absorbed by the plant and some light is reflected. So the absorption and reflectance, it depends upon the physical and chemical characters of the plant. So since the chemical characters affect this reflectance, so if we measure the reflectance, we will know what is the chemical composition. So that units are highly useful to measure what is the nitrogen status of the plant, what is the carbohydrate status of the plant, how much sugar the plant produce, how much nitrogen the plant tissue has. So this is very important to quantify the biochemical characters without doing the conventional method of laboratory analysis.
And this is all non-destructive. So the plant remains alive after doing all this.
Yeah, this is all non-destructive, and it is also faster. Otherwise, conventional method for each of the analysis, it takes 2 to 3 days. So the throughput is less, and the plant, we need not destroy the plant, and we can do more analysis, and we can, importantly, we can do repeatedly the same plant can be imaged at different growth stages, which is not possible in the conventional method.
Let's go and see the imaging part of this. So are these all the camera units which are taking pictures?
Yes, each chamber consisting of different kind of camera that I have explained. So the images are taken inside these units.
So this is how it would look like.
This is the chlorophyll fluorescence imaging. So this is the normal plant, how it looks like, and this is under the drought stress condition. In these two plants are there. So one plant still it has maintained its chlorophyll fluorescence, and the other plant is completely gone. So we can easily identify the tolerant plant. By visual it is very difficult.
And how did you normally do before you had this center?
Say if you want to know the biomass, we have to cut the plant and we have to dry it in the oven, and then we can take the weight manually. See, it takes about 2 to 3 days. If you want to measure nitrogen content, even it takes more time. After drying the plant, we have to grind, then we have to do chemical processing. It takes more than a week. So that is done within a minute in this facility.
And the plant would get destroyed in that.
Yeah, in that plant get destroyed. Here the plant is not destroyed. It is the repeated imaging can be possible.
What do we see here and what happens here, sir, for this plant?
So this is weighing and watering station. It measures the amount of water used by the plant. So it weighs, you can see the weight of the plant here. And if the weight is less, if water is exhausted, then the irrigation will be given and then the plants moves to its greenhouse.
Let's go and see the imaging part, sir. In this highly automated facility, imaging is what is important. What do we see here in this image and what does it represent, sir?
So as I explained there, each imaging unit is has a different sensor. What we see here is the image from the chlorophyll fluorescence imaging camera. So this takes image of photosynthesis system, how active the photosynthesis system is there. So we can see the green color, dark green color indicates the plant leaves are under healthy condition. If you see in the red color, this shows that the photosystem is damaged. So this gives out of the total pixel, how many pixels of the plant or how many leaves of the plants are in good condition with reference to the drought treatment that has been given to this plant.
And will this be able to indicate to you whether this plant can tolerate and become useful in a climate changing scenario?
Yes, this is very important character of the plant to maintain the photosynthetic ability. So this indicates clearly whether the plant is tolerant or sensitive. So we take the images of all the germplasm line and the plants which maintain more of this green color or more of the photosystem intactness, we select those plants.
Dr. Vishwanathan, the father of India's green revolution, Professor M.S. Swaminathan visited this facility. What did he say in short?
So Professor M.S. Swaminathan highly appreciated this facility and he hoped that this facility will be helping in developing climate resilient crop varieties.
Let us now listen to what Dr. M.S. Swaminathan had to say when he visited this facility and he came on a wheelchair.
I am extremely happy to be here today to see this wonderful facility. I think when I was young and student, we had so much trouble in recording accurate data, particularly in relation to phenotype, because phenotype is a product of the interaction between genotype and the environment. And the environment has many, many parameters. Here in this facility, beautiful facility, and I'm happy it is dedicated to Nanaji Deshmukh, whom I had known well, and who wanted the best in science to go to the poorest of the poor, the villages. And that is the purpose also of IARI and ICR, to take the best in modern technology to small and marginal farmers. I hope this facility will be used not only to do a lot of theoretical work, but also in developing new kinds of phenotypes, which will help our farmers to produce more at less cost.
Having seen this imaging inside the facility, similar stuff is also done in the field using very similar sensors. Let's go and see that. Dr. Jinaswamy, this technology which is used for phenomics, you are also using in the field with drones and small mobiles?
Yes, what we have seen inside the facility, all the sensors are built in the sensor room and the plants move to the camera and the images are taken. Here all the sensor can be put in the drone as well as in the mobile platform for taking images. So for example, in this drone we can use the visual sensor which take the color image. Based on that we can know how is the crop growth, biomass, and in this drone we can add thermal camera which tells about the plant water status and what is the transpiration rate which we have seen inside, and we can use either multispectral or hyperspectral data. It will give about the health data, that is how much nitrogen is there, what is the plant health. So we can get the health data of the plant, and it can also measure various stresses, either it is the effect of abiotic stresses such as drought or high temperature or salinity. So that information can be captured, how the genotype different, and we can also capture the information about pest and diseases. That is, if the pest started infecting, immediately it induces some changes in the plant. Same way the pathogens start infecting, immediately it will induce some changes. So before we see visually in the large scale area, we can detect early by using this technology. So we can take immediate action, and we can take the control measure only within that particular area. So this one is very useful for phenotyping, wherein we can phenotype, if we have a large number of germplasm, we can put each genotype in few rows, and then we can collect the phenotype information. And also we can use this technology for precision agriculture, which will be a great application which is coming in near future. So wherein we can use the drone to monitor the plant health and apply either input or the agrochemicals by using variable rate technology.
Do you also do, using this mobile cart, some similar work?
Yes, in the mobile carts also we can use the same kind of sensor what we have put in the drone. In addition to the imaging sensor in this platform, we can also use spectroradiometer and other sensors that are based on the remote sensing method. So we can use all the cameras that are seen in the Phenomens facility, as well as in addition to the radiometric sensor can also be deployed. So in this advantage is that you can take a more closer look of the plant.
Now we are seeing mustard. Can it be done for other crops?
Yeah, this technology is useful for any crop, even for all the field crop, even for the horticultural crop, where going inside and taking observation is very difficult. So it can be highly useful for horticultural crop, such as the coffee or tea estate, where daily going and monitoring the plant will be very difficult.
Thanks a lot, Dr. Vishwanathan, for explaining to us the complexities of phenomics and understanding the genotypes of plants in a climate changing regime. Thanks a lot, sir.
Thank you so much. Thank you.
Trying to understand how plants behave in a changing climate regime and how plants behave as the world is warming up is critical for feeding India as the world warms up. On this episode of Life and Science with Pallav Bagla, we saw the whole body scans in a non-invasive way of plants, and also using drones and mobile platforms for scanning fields, all in an effort to prepare India for a climate resilient agriculture. Precision agriculture at its best. Smart agriculture in times to come. Keep watching India Science.